Hi everybody. So I guess you can recognize my voice, right? Yep, this is your lecturer, Youngtae Chang. And today's lecture is the structure of cells. For this course, I could choose the different references or textbook, but we are going to try as much as possible just the free references. So in this case, I'm going to designate some of the Wikipedia with the following titles: Cell and Human Body. So you can easily find those reading material from website, right? And in this lecture, so the title is the structure of a cell. I wish you can learn the component of cells and their function, and also to figure out how they are connected to each other and how they are dynamic, and also how do we define the in and out of our body. There are many different life forms on Earth. One of those very popular other model, the live cell, is this, the onions. Maybe partially because those onion cells easy to get, and also it's easy to monitor or observe different stages of those cells. In this picture, now we can see here the A cells. These cells are interphase. They are kind of normal status. And B here, now you can see those nucleus is condensing and they are preparing for cell division. And here C, actually the cells are dividing and the condensed chromosome is aligned in the center of the cells. After this aligning and cell division is fully done, now we can see here two cells are separated and new cell wall is forming up. Now we call these two cells are the daughter cells. So here we can see one snapshot of the different other stages of cells using onion as a model. Now we are moving to more dynamic other features of those cells. Follow me. Here is beautiful movie for the cells. Cell has a different structures inside, and usually cell has the lipid membrane outside to segregate its content from environment. Inside the cell, we call this the space the cytoplasm. And then if you look at those cell structure, and there are many different kind of small organelles inside. And in the center, there is the DNA containing nucleus. And DNA is the genetic materials. The cells on Earth, in big classifications, we can say one is the eukaryotic cell, and eukaryotic cell has much more complicated organelle structure, but most important thing is the nucleus in the center. So, eukaryotic cell has a DNA inside the separate membrane structure of a nucleus. Most of those advanced cells, such as the plant cells or animal cells, our cell too, and this cell has a nucleus, so all of these advanced cells we call eukaryotic cells. In comparison, prokaryotic cells, it's a more primitive the cell shape. It does have a DNA, the genetic information, but it doesn't have separate the nuclear structure, so the DNA structure is spread out to the cells. Comparing to eukaryotic cells, this prokaryotic cells is much more the simpler structure, and most of those the unicellular organisms such as bacteria is belonging to prokaryotic cells. Organelle means little organs, and inside the cell there are many the different type of organelles, especially in eukaryotic cells. They are complicated and also many different type of small organelles. And first of all, in the center, there's a nucleus, which contains the genetic information of DNA. And outside of this nucleus, there is intracellular membrane structure, so-called endoplasmic reticulum. Abbreviated, we call it ER. Outside of ER, another connected the membrane structure, this is Golgi body. This Golgi body was found or discovered by Dr. Golgi, that's why we call it Golgi body. And also, there is another structure, mitochondria, and this kind of small vesicles, including lysosomes. Let's look at the nucleus first. nucleus here, through those nuclear pore, inside the nucleus, there are lots of DNAs. DNA is a genetic information, and this DNA in the interstate, they are formed, they exist as a chromatin structure, and then it is segregated from the cytoplasm using nuclear membrane. When cell aggregate, this chromatin structure aggregated together to form highly condensed chromosome form, and utilizing this chromosome and sharing genetic information into two different daughter cells is possible. In the nucleus, there is another structure so-called the nucleolus. Nucleolus is a synthetic center for ribosome. Ribosome is composed of ribosomal RNA. Once the ribosome is synthesized, they are coming out of the nucleus, and then the ribosome is the center of the protein synthesis. Depending on those proteins' final destination, some of those ribosome is existing in cytoplasm area, and then the product of that protein reside inside the cells. But some ribosomes, they are attached to the endoplasmic reticulum structure, and a lot of ribosome existing on the EL, and the surface looks like very tough. So that's why this area is called as a rough endoplasmic reticulum. Some other structures, they have a smaller ribosome structure. That's why they are called as smooth EL. If the protein is synthesized inside this the endoplasmic reticulum, this protein is transported to the outside, and this is the membrane structure of EL. If you look at carefully, now this is ER structure. Protein was synthesized inside here, and then they are moving out of this EL, but it's not like individual protein. They are encapsulated in this vesicle, and then this vesicle is moving out of the ERs. So this is like a spatial shuttle for proteins, and those vesicles are moving to next membrane structures, the so-called Golgi body. And inside the Golgi body, now this proteins is getting matured, and first of all, they can get the folding, proper folding inside the Golgi body. And a lot of modification can also happen here. Sometimes the lipid molecule can be added here, or sometimes carbohydrate structure is also attached to the protein. This carbohydrate modification we call glycosylations. This lipidation or glycosylation, these are part of post-translational modifications. In cytoplasm area, there are other vesicles so-called lysosome. Lysosome can swallow many trace materials in the cell, and using acidic environment and lots of digestive enzymes, they can destroy them. Cell has another very interesting type of organelles, so-called mitochondria. Mitochondria is the center of cellular respirations. When you take food, those nutrients is digested, and some of those components getting into the mitochondria, and it is converted into biological energy, ATPs. Very interestingly, this mitochondria is not originated from our own body. Seems like in ancient time, mitochondria was separate life forms, and when they entered into our body, they took up this very unique and very important role of cellular respiration, and then became part of our body. And this is a perfect example of symbiosis. So that's why mitochondria has its own DNA and its own genomes. To maintain the cell structures, inside the cell there is another protein, the so-called cytoskeleton. In cytoskeleton there are microfilaments and also microtubules and other proteins. Using these cytoskeleton structures, we can form our body shape and cell shape. Different life form has a different shape and a different type of cells. If you look at those green color of leaves, and inside those leaves, of course, there are cells. These are plant cells. Plant cells has a very unique structure so-called chloroplast. Chloroplast has green pigmented material so-called chlorophyll, and this chlorophyll is the center of photosynthesis. And plant cells has a very unique structure outside, so-called cell wall. Animal cells has only cell membrane, and we don't have other cell wall. Because of the cell wall, they can keep much rigid the structure of those plant cells. Bacteria has a very unique another structure, so-called flagella. Using this flagella, bacteria can swim in the water or maybe in our body fluid. We have a similar type of cells like this. Yes, it's a sperm cells. At least shape-wise, we have only one flagella, but using this flagella, we can swim around. We have seen different type of organelles in our cells. And if you look at the cells again, so this is a cell membrane, and this is outside of cells, and this is inside of cells. This picture was drawn by the scientist David Goodshell, and this is representing quite important point. Usually we can think the cell is just one bag, and inside the cell it's like just liquid, the material, so-called the cytoplasm, and then those organelles are just hang around here. But in reality, actually inside this cell, the mass of those material is huge. The material amount inside the cell, if you calculate, it's about 300 to 400 milligram per milliliter concentrations. Can you guess how dense they are? Okay, here, let's take one quiz. Let's assume that the molecular weight of this macromolecule is 10,000. And if the amount of those material is 300 milligram per ml, what is the molar concentration of that macromolecule? Let me say again, molecular weight is 10,000. Amount is 300 milligram per ml. Could you calculate those molar concentration of those material? I will stop the video here, and you calculate and go to poll and answer your questions. Did you get the answer? Yes. Approximately the concentration is a 30 millimolar concentration. If the molecular weight is 1000, it becomes 300 millimolar. If the molecular weight is 200, near to the glucose is 180, now the concentration becomes more than 1 molar concentrations. It's an amazingly high the concentration, and you should keep in mind, inside the cell, it's a highly crowded environment. It's not like just a fish swimming in the water. It's more like the turtle is crawling in the mud kind of a situation. That's the real environment inside our cells. Okay, let's come back to the cell structures. This is a nuclear pores, and the synthesized ribosome and mRNA is exported to the cytoplasm. And in the cytoplasm, there's machineries coming and binding to this the mRNA. Now this structure is bound to two structures together, right? This is ribosome, and then reading this mRNA, it starts to synthesize the proteins. This is a ribosome structure, and this is an mRNA. And by reading this mRNA, sliding here, right? And then synthesizing proteins behind here. So by reading this mRNA, those are the synthesized protein is secreted inside endoplasmic reticulum. Interesting, right? So now this is mRNA. Synthesized protein is secreted into the endoplasmic reticulum, and this endoplasmic reticulum surface, now they form these vesicles we have seen before, and this vesicle is moving to Golgi. But this vesicle is not flying to the Golgi. Actually, this vesicle is walking along this tube, so-called microtubule structure, utilizing these motor proteins. So using this the motor protein, now the vesicle is transported to the Golgi structure, this Golgi body. And inside the Golgi, this vesicle is fused into Golgi body, And then protein folding and post-translational modification, all those things are happening here. After Golgi, it is further moving. This vesicles is again using this motor protein, and along this microtubule structure, it is moving to plasma membrane. In the plasma membrane, so-called through exocytosis, the membrane fusion, now these proteins are exposed to the outside of the cell. Many proteins are plasma membrane-bound proteins. P-selectin is one of them. When leukocyte is rolling on the blood vessels, they utilize this P-selectin interaction to each other. And if you look at those bloodstream, this red blood cell is flowing very fast, but leukocytes is relatively slower, and then they are not flowing, they are rolling on the surface of this blood vessel. In this case, they utilize this P-selectin as a binding motif, and when they find this inflamed area, they can enter into the tissues. If you look at those P-selectin, it's not single molecule to single molecule interaction. As you can see here, it's a multivalent interactions. That's why even one binding is not strong enough. Into cell-to-cell interactions, there are multivalent interactions happening. Now this is the membrane structure, the watching from outside of cells. Some of those areas, very interestingly, this is membrane structure, and some area is called as very rigid structure, so-called lipid raft area. On this lipid raft area, there are the proteins, the receptors are embedded inside here, and it looks like it's a raft floating on the river like this. This lipid raft structure has a rigid structure because of this cholesterol other components inside here. So this is outside of the cell, this is inside of the cell, and this is a lipid bilayer. Other area has more flexible other phospholipid, but this area has high contents of cholesterol, and that makes this the lipid part is much more rigid than other parts. And utilizing this rigid structure, receptors can have anchor here and then bound in this area. Outside of cells, there's a lot of these signaling molecules, so-called chemokine, and its partner of receptor is waiting for the signal on the plasma membrane. And usually these receptors are located in this the rigid part of those membrane. If you look beneath of this membrane, now this is inside the cells, you can see this lipid raft floating around. And these proteins, sometimes this is a transmembrane protein, or this is anchored through this the lipid molecules. This is a protein and this is the lipid molecules. And inside the cells, there is another cytoskeletal network here. This network is so-called spectrin structures in tetramer forms, and they make this kind of a network right beneath the membrane to make those membrane has the rigid structures. Across this spectrin network, there are many proteins are embedded in the membrane, connecting the signal from outside to inside. Now, if you go deeper inside the cells, there's another the cytoskeletal structure, so-called actin filaments, is forming up those strong network inside the cell. Utilizing this actin filament, cell can maintain the shape of those body. And also, this actin filament is highly dynamic. This is a very important component for cell moving. If the cell is moving to one direction, then the moving end part should have the very fast the formation of a filament, but the other end should cleave and the disassemble of those actin structure. Here, the severin protein is acting that kind of role. Another important cytoskeletal structure is microtubule. As the name implies, this structure has this kind of tubular structure, and it has two ends. One end is the very fast assembly and moving forward. This is a plus end. And the other side of this end is very fast for the degradation. And that's why this is called as minus end. Of course, both sides can assemble and disassemble, but speed-wise, plus the end is growing side and minus the end is the disassembly side. As we have seen before, this is basic movement happening on these microtubule structures. So that's why we can call this microtubule as like a railroad of the cells. And this motor protein, in this case, this is a dynein, which has the long neck and then 2 feet. That's quite cute, isn't it? And this microtubule structure, utilizing this microtubule structure, not only these vesicles, another important organelle we have seen before, this kind of structure, mitochondria, is also moving. It's like a circus, the moving along those lines, right? So all of this microtubule is serving that kind of a network. Inside the cell, there is a structure so-called the centrosome. This will be the center for cell division later, and all the microtubule structures are connected to this centrosome, and that's why we can say those direction. One direction is from inside to outside, and the other one is outside to inside. Okay, how was the movie? So now we came back to a simple model here. So in this picture, we have a cell model, and here we have a cell membrane and nucleus, and this is the endoplasmic reticulum, and this is Golgi, and this is mitochondria, and some the small vesicles around here. Now let's think one problem. Now here, this is cell membrane, right? And then can you say which one is inside the cell or outside of the cells? Maybe for cells, sounds like it's an easy problem. Then can you think that what's the difference among those components inside the cell and outside the cells? What kind of molecules or ions is more abundant inside the cell, and which one is more abundant in outside of cells? And also, why they are?